Hello, everyone, and welcome to today's keynote webinar titled Maximizing the Potential of 3D Organoid Research with Automated Isolation. This webinar is a part of the Future of Biomedicine, Reproducible Advanced Cell Systems, and Translational Insights. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by Sartorius. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. And with that, I'd like to now present today's speaker, Andris Abramenkovs, Product Manager, Cell Selector at Sartorius. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Andris, you may now begin your presentation.
Hello, everyone, and thank you for joining today's session, where we will be discussing how to maximize the potential of 3D organoid research using automated solutions. 2D cell cultures are extensively utilized in biomedical research. However, they lack the complexity of actual tissues, resulting in oversimplified models that might not accurately depict the progression of disease or response to a treatment. Cells in one layer have limited opportunities for cell-to-cell interactions, which is a significant drawback as these interactions are crucial in many biological processes. It is important to note that drugs often behave differently in 2D cultures in comparison to more advanced models. This discrepancy can lead to inaccurate predictions about drugs' efficacy and safety, potentially impacting the drug development process and associated costs. To help overcome some of the challenges associated with 2D cell cultures, organoid models have been developed. But what exactly are organoids? Essentially, they are miniaturized and simplified versions of organs. These three-dimensional structures mimic the complexity and functionality of actual organs. They offer unique insights into intricate interactions and processes within biological systems, which were previously impossible to study in such a detail. The key advantage of organoids is their complex structure, which is more representative of the in vivo environment. This means they can better mimic the intricate interactions between different cell types and the extracellular matrix found in actual tissues. Organoids can also more accurately model disease progression and response to treatment due to their realistic cell interaction and the structure. Therefore, organoids provide a better estimate of drug efficacy and toxicity as they closely mimic the in vivo environment, where they can offer also more accurate prediction of how the drug will behave in the body in comparison to a 2D model. Therefore, organoids can help to streamline the drug development process and increase the likelihood of clinical success. By replicating the complex interactions between different cell types and extracellular matrix, organoids can provide a more accurate representation of how disease progresses in the body. This leads to a better understanding of disease mechanisms and potentially aids in the development of more effective treatments. So what are the sources of cells for the organoid culture? First, we can use embryonic bodies, which contains embryonic stem cells. And once these cells are cultured in a 3D culture, they spontaneously form certain three-dimensional structures. And upon addition of extracellular matrix, they mature into an organoids. When working with embryonic cells, there are some legal considerations as well as some ethical ones. Therefore, most oftenly, the cells for organoid culture come from patients or healthy donor. So there's different options that we can utilize. We can take somatic cells, reprogram them into induced pluripotent stem cells, and then culture them again in a 3D structure. Alternatively, we can also take cells from normal tissue and extract adult stem cells that again will form this 3D structure, and upon addition of extracellular matrix, we get an organoid culture. As well, we can also derive organoids from, for example, diseased patients. For example, cancer tissue, we take out the cancer cells, add the extracellular matrix, and we generate a 3D structure that is more often referred as a tumoroid. Organoid research is a rapidly evolving field that is gaining momentum due to its wide range of applications. Organoids can be used to study organ development, model diseases, test drugs, and even contribute to the fields of regenerative and personalized medicine. One significant advantage of organoid research is its potential to reduce our reliance on animal models. Traditional animal models, while useful, often fail to accurately present human biology. Organoids derived from human cells can provide more accurate and ethical alternative for studying human disease and testing new treatments. However, like with any pioneering field, the organoid system presents unique challenges. So what are some of these challenges? Organoids are technically more complex than traditional 2D cell cultures, and therefore they necessitate a deeper understanding of cell biology and more sophisticated techniques for culture and maintenance. This complexity can present a steep learning curve for researchers new to the field. And organoids typically require more expensive materials for culturing and extended culturing times. This can increase the costs and duration of experiments, potentially limiting the testing of multiple conditions. Also, analyzing organoids can be challenging and often requires more sophisticated acquisition and analysis solution. Unlike 2D cell cultures, which can be easily observed under a microscope, organoids are three-dimensional structures that require more advanced imaging techniques for visualization, selection, and analyses. Due to the complex nature of organoids, producing consistent results across different experiments can be difficult. This lack of standardization can make it challenging to compare results and draw definite conclusions. Therefore, additionally, data analyses of organoid assays are needed and can be time-consuming. Given the complexity, it often takes longer to perform experiments and analyze these results. Now let's talk a little bit more about the workflows focusing on drug treatment testing using organoids. The drug testing workflow usually involves several steps. First, we need to grow and expand our organoids. Then we need to transfer the organoids to a plate that is compatible with compound screening, such as, for example, 96-fold plates. Then we need to treat the cells and monitor their response, which can include changes in organoid size, shape, or function, as well as more specific molecular changes, such as gene expression or protein activity. Despite the potential of organoids in drug testing, there are several challenges that need to be addressed. One of the primary challenges is the variability between the organoids, which can generate inconsistent results. Therefore, it's critical to monitor organoid growth, morphology, and behavior, as well as determine the optimal time for organoid transfer before testing the compounds. Transferring organoids is not a trivial task, as it usually is a manual process, which drastically affects the throughput and viability. Also, isolating and depositing organoids from biomatrices is not as easy, as biomatrices are temperature sensitive and challenging to work with. Usually, this also affects the throughput of the assay and in some cases also the viability of organoids. Once we have successfully transferred the organoids and added the drugs, we need to monitor the response of the organoids again, but now in a high throughput manner. So how we can overcome these challenges? Organoid research and workflows can significantly benefit from automation. Automated imaging systems, for instance, can continuously monitor organoid growth, provide real-time data on changes in size, shape, structure, and behaviors of the organoids. This not only allows for more precise tracking of the organoid development, but also enables early detection of any abnormalities. Screening various compounds can be extremely expensive, so detecting any deviations from normal growth profiles can save a significant amount of materials and costs. When it comes to organoid isolation and transfer, automation can increase throughput and organoid viability while providing proper documentation and characterization of transferred organoids. Sartorius offers IncuSight, a live cell imaging platform for monitoring organoid response to treatments and also monitor the cell and organoid growth. As well, Sartorius offers automated organoid isolation system called Cell Selector Flex, and we will be mainly focusing for the rest of the speech today on this specific platform. Cell Selector is an automated cell isolation platform that consists of precise robotics arm that can retrieve and transfer cells from source vessel to the destination plate. It also contains an automated microscope with bright field, phase contrast, and extended fluorescence capabilities. The instrument is connected with a powerful workstation that allows to control the system as well as analyze and document the results. The Cell Selector platform are a customizable system that can be configured to meet your research needs. Let's take a closer look at the process of identification and isolation of organoids using Cell Selector. We start by first identification of 3D structures, where we can leverage multiple imaging modalities such as fluorescence and bright field imaging. The heterogeneity of organoids can complicate result interpretation after drug treatment, as organoids can significantly vary in their size, shape, and overall 3D structure. Therefore, using Cell Selector during the analyses, we have the possibility to select and filter using multiple parameters such as size, sphericity, object darkness, radius, and fluorescence intensity. It greatly aids in quick identification and isolation of organoids with similar properties. Additionally, the ability to quickly review filtered structures just before isolation ensures that the selection process has been done correctly. This is particularly vital when isolating a large number of organoids for high-throughput screening assays. When it comes to the picking, Cell Selector Flex platform features exchangeable robotic arms that can extend the range of retrieved organoid structures. Single-cell picking module allows to isolate smaller organoids of sizes between 80 to 220 micrometers. For the retrieval process, here we are using reusable glass capillaries. For larger organoids, we can use a semi-solid media picking module that allows to pick between 500 micrometers to 3.5 millimeters of size. It is possible here to use disposable plastic tips that can be automatically exchanged between the picks. Most organoids are grown in biomatrix, which complicates the isolation process as the biomatrices at physiological temperatures are gel-like substances. In the top right panel, you can see a successful picking of organoids from biomatrix using Cell Selector Flex platform. Images before and after picking are shown. It is also important when picking organoids from biomatrix that the process does not disturb surrounding structures or critically damage the biomatrix itself. In the bottom right panel, you can see an example of organoid retrieval without perturbing surrounding organoids. Additionally, organoid isolation has to be a quick process so that organoid viability is not impacted by populating of multi-well plates. If we are extracting organoids from suspension, then we need to consider the movement of the organoids, which usually occurs slightly during imaging. Having an automated solution that automatically detects and corrects for this movement is essential. The top right panel is an example of organoid drift and automatic detection of organoid movement during the imaging of suspension organoids. This process can be automatically done within the Cell Selector software. Importantly, the picking process also must preserve the organoid morphology and viability, as affecting these parameters will significantly impact the organoid response in drug screening. The bottom right panel shows unchanged morphology of organoids before and after picking using Cell Selector Flex platform. Using Cell Selector Flex, organoid picking is a robust process where we can pick organoids from various formats of vessels. We can also pick various types of structures of the organoids without perturbing the structure itself. As an examples of successful picking, here you can see an isolation of human endometrial organoids, human intestinal, lung, retinal, colon, or heart organoids. Here we have isolated them from either ultra-low attachment plates or biomatrix. Once an organoid is picked, it needs to be successfully deposited in the destination plate. If the organoids are to be placed in a biomatrix, then the destination plates on a cell selector can be cooled during the organoid transfer process to prevent any premature polymerization of biomatrix, which could ruin the entire experiment. Dispensing organoids in a biomatrix can produce bubbles if the process is not tightly controlled. So leaving the bubble in a biomatrix during deposition will create a lower density region where the organoid can expand more easily, affecting its morphology. In the panel on the right, you can see microscopic and microscopic images of the deposited organoids in a biomatrix in a multi-well plates using cell selector. Upon a closer inspection, we can see that there are no bubbles after the transfer of the organoids, and the morphology is looking healthy. Once we have successfully completed the experiment, we can generate the proper documentation for each step of the process. Starting from overview images, quality control images showing successful picking, and a summary table with picked object properties, IDs, and destination plate positions. Now I would like to summarize the benefits of using Cell Selector Flex for organoid isolation. Cell Selector Flex allows to automatically scan, detect, gate complex 3D structures based on their morphological and fluorescence parameters, which is essential for high throughput experiments. Without such capabilities, we would not be able to increase the throughput. Gentle transfer of wide range of organoid sizes from and to biomatrices allows us to completely replace manual process and focus on the reproducibility and robustness of the data that is required for further analyses. Rapid transfer of organoids using automated solution allows us to increase the reproducibility as well as the viability of our organoids. This also prevents them spending a lot of time outside their natural environment, which can potentially affect their morphology as well. Organoid transfer with exceptionally low volumes allows us to inject only desired material in a biomatrix without generating any additional bubbles, which means that we can completely control the process and move on to our drug testing without worrying about morphological changes that can potentially happen after the transfer. And once we have actually completed the whole process, we have the possibilities to extract full report for the documentation purposes. In conclusion, Cell Selector platform can simplify organoid expansion for high throughput assays. Automation also provides more consistent and reproducible results of organoid transfer process, as well as the imaging. Automatization of organoid culture workflows can reduce time and resources required for making organoid cultures more cost-effective. Cell Selector platform allows quick identification and transfer of organoids while providing proper documentation in the process. Please reach out to us to learn how we can help you to simplify your organoid workflows, or visit sartorius.com today. With that, I would like to thank you for tuning in today, and I would be more than happy to take any questions that you might have.
All right. Well, thank you so much, Andris, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Submit your questions in the Ask a Question box located on the bottom of your screen. All right, so let's get started. We've already got a few questions in here. Andris, this first question asks, is it possible to select organoids using multiple fluorescence channels?
Yes, there is a possibility to select multiple fluorescent channels in addition to morphological parameters using a cell selector platform, as these kind of features are actually inbuilt in the software itself.
Great, thank you. All right, next question here. Is it possible to pool multiple organoids in one well using cell selector?
Yeah, that's a good question. Actually, using cell selector, we can actually take and select multiple organoids from one source vessel and put it in one destination well. And there are some advantages actually using this kind of approach when we are thinking about high throughput analyses of different type of drug treatments. We can just select organoids with similar sizes and properties, add our compounds, and monitor how these very, very similar organoids are responding to the treatment, and if the response is very homogeneous.
Great, thank you. All right, and a reminder to our audience that if you would like to have any questions answered, please submit those now. This next question that we have here, from what kind of biomatrices organoids can be isolated?
So here we can actually isolate organisms from almost any type of biomatrix, as long as we have some transparency and we can see that there's no interference between imaging and actually selection of the organoids themselves. So here we're talking about matrigel, hydrogel, low concentration collagen, for example, etc.
Great, thank you. All right, another question we have here. What are the plate formats compatible with cell selector?
So regarding the source plates, we can use various type of formats, even custom formats. So we can use a standard 96 well plate, 6 well plates, Petri dishes, 96, 384 well plate formats, almost anything that you would like to, there's a possibilities to pick from. And when it comes to destination plates, we can again deposit the cells into a 96 well plates, 384 well plates. We can use even Petri dishes or custom formats as well here.
Wonderful, thank you. All right. Okay, well, we are going to wrap up with that final question there. So Andris, I want to thank you again for joining us today. Do you have any final comments for our audience?
Thank you for all the questions. And if you have any further information that you require, please do not hesitate to reach out to us, and we'll be more than happy to help you.
Wonderful. Well, thank you again, Andris, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until January 23rd, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.